Excellent, and this will be available on the website afterwards. And let's get started. It's 2 minutes past. My intro is not as important as the presentation, so when I get started. All right. So I want to welcome everyone to our summer webinar, the first of our summer webinars. And today we have 3 speakers, including Maria, which I failed to put your picture on here, Maria. I'm sorry. So today's webinar is going to be one of the more exciting ones from my perspective. We're going to hear some novel results and about a novel method from Jason Planick, who many of you know, and Patrick Smadbeck. And they're going to be talking about this new method that we've had in the portal for quite some time, novel method that is. And then Maria is going to take over and talk about sort of all of the new production release highlights that have come as of June this year. So I'm going to start with a little brief context as to what the presentation, the scientific presentation you're going to hear now matters. And I'll come at it from my perspective real quick, because I think this is one of the things that sets our resource apart from so many of the others out there. And one is, so many resources do a great job of representing the mature GWAS data sets that are out there now. And they're large in number. There are thousands of data sets from multiple traits, many traits in many cases now. And many of them do a great job of finding these, right? This is something we've done well as a field for complex trait genetics. Our knowledge portal itself has done a great job for this, in my opinion. We started with just being able to, this is back in late 2019, where you would be able to type in your favorite gene and get a list of the common variants, but you would see the largest data set represented to you. But you can imagine for many of this, for this diabetes portal, and now the common metabolic disease knowledge portal, we have a lot of data sets for a given trait. For example, for BMI, there are 84 data sets in the knowledge portal today. So that means that in many cases, there are unique samples in different data sets, or in many, many cases, a lot of these data sets overlap, because our colleagues in this field are doing meta-analyses that are incorporating many of these data. So that's why we always presented the largest meta-analysis on the portal. But in many cases, there are samples that we are losing as a result of not incorporating all the data that are out there for BMI or these other traits. So what we're very lucky is that, we are now moving to a world where if you type in your favorite gene now, you can see the distilled results for a given trait. Here's BMI for FTO, for example, another view of that. And you see it for all of the other traits in the portal. And how is this coming to be? Well, we used to show you the largest data set. Now we show you an incorporated distilled results, a meta-analysis that incorporates sample overlap and presents to you the top signals that we find novelly, essentially in the portal, based on all the data that's out there, that's already been published, but also the integration of all those data sets across the portal. So why does this matter? In summary, no other resource presents meta-analyses results for its data. GWAS catalog doesn't do it. The other large genome-wide association study resources don't do that. So that's what's different about us. Another one, no resource aims to account for these sample overlaps in many of the studies, because in many cases, there's almost 100% overlap in data sets that are presented on so many of these resources. And finally, I think it's aspirational, but no other resource distills these results and signals for you for a non-expert user. If you're a GWAS catalog user, you have to know a little bit about genetics. Our portal tends to shift you away from, if you're not an expert geneticist, how might you navigate all these signals for all these traits? And that's really want to get to with the portal. And this method you're going to hear results of today really aims to start to do some of those things. So I'm going to turn over to Jason and Patrick for the presentation.
All right, excellent. All right, so we are really, it's been a long time coming, I think, for us to finally share some of these results we'll talk about today. The bottom line method is probably the one that we get the most questions about on the portal, how it's implemented, what exactly it's doing, is it valid? And hopefully we will be able to answer some of those questions today. I'm going to talk through just the setup. Patrick will talk through how we implement it in detail and some of the statistics underlying it. And then I'll show some results on kind of evaluating how good these are. And so the first thing I just want to make sure to say is that the bottom line statistical method was initially developed by Sivanti Gupta with Gonzalo Avacasis and Mike Benke at Michigan. So so the statistics Patrick will be covering are a product of them. The analysis I'll be showing have been done chiefly by freq and Biokol, who's a undergraduate student in our group, and then in addition to him, some help from Monica Ruiz, who was a visiting PhD student in our group. So just I'll be presenting work from them. Okay, so as Noel mentioned, the goal of the bottom line method is to go from a table like this, where you have for a given trait, in this case diabetes, numerous data sets, numerous p-values for any given variant, it's totally heterogeneous data, into this, which is a single estimation of association strength between that variant and every trait in the portal. And obviously the challenge is that if you only have a single data set for a trait, it's easy, right? You just look up the p-value in that data set. But it gets tricky once you have multiple data sets. And in particular, in some cases, data sets that we have no idea whether they share samples or whether they're the same or different. And these are all the challenges that we're dealing with, only being presented with summary statistics, how to combine them. So there's actually three ways that you might immediately come to mind if we presented you with this problem. So here's a toy example where these are p-values for a fictitious variant. It's observed in 4 different data sets. So this is all for the same trait. The sample sizes say range from 10,000 to 100,000, and the variant has different p-values. So it's very strongly associated in the data set that has 10,000 samples. It's kind of associated in the data set that has 30, not at all in the data set that is 50, and kind of intermediate in the data set that is 100,000. So what p-value and effect size should we report for this variant? So the first thing that you might do would be to report the p-value that is the most significant. So across all the data sets, the strongest association. So depending on your background, whether you're a statistical person or a biologist, this may strike you as totally reasonable or totally unreasonable. But I want to emphasize that this is what actually, by far and away, the most widely used integration method in the literature. If you're searching a catalog and you're looking for all the associations, say, the GWAS catalog has, and you consider a variant associated, if it's in the catalog, you're using the minimum p-value method, right? If you are mining the literature and you see a report of an association and you use that to then functionally characterize like that variant, you're using the minimum p-value, because the minimum p-value is equivalent to considering a variant associated if it has association in any study that's ever been published. So it's actually whether or not you think this is reasonable, it's very, very widely used. And in fact, the default, I think, that are used really implicitly in many cases throughout the literature as well. Now, obviously, the issue with this is that it's not a statistically valid analysis. If you formally do this analysis across all of the variants for a given trait, you see massive inflation. So this is a QQ plot of what happens when you take the minimum p-value for BMI associations, and you see, for those who are familiar with lambda values, an outrageous value, and clearly a statistic that's not well calibrated. So an alternative going in the other direction would be to take the association from the largest data set, right? So we don't take the p-value based on the most significant, but we take the one that's the largest under the assumption that has the most power, and that's the most recent study in most cases. This is the best if you really want like a curated analysis that somebody has picked over and an expert has vetted, is probably the best that you can do. And I would note that this is also probably your best shot if you want to do post-processing analyses of the GWAS, like credible set analysis or LD score regression, because you want to start with a data set that has really been curated. But the issue with this is that, as Noel mentioned, are we throwing away information, right? So maybe we take the p-value from this top data set for BMI here that has 484,000 samples. That would mean throwing away, say, this Biobank Japan GWAS that has 158,000 samples, which is smaller than the largest, but still information that we probably don't want to just discard without thinking about it. So the formal statistical way that you combine data sets usually is through meta-analysis, where you essentially combine all of the studies together and you weight them according to their sample size or their standard error. And this is to produce a p-value that kind of adequately summarizes across all of the studies statistically. And this is what everyone does. Obviously, this is the best practice approach. The largest giant GWAS and the largest BMI GWAS in the portal, and most of those actually are meta-analyses. This one in particular was a meta-analysis of a 250,000 sample study and a 450,000 sample study And this is obviously the statistically principled thing to do. Now, the issue, though, is that it's only principled when the studies do not overlap. And so in some cases, we can remove studies that obviously overlap. So you can probably have been showing this table, guess that the giant UK biobank results overlap with the male subset of that. But in other cases, not so obvious, right? So giant is mostly European, so it probably doesn't overlap with the biobank Japan GWAS, but it very well may contain samples from Godarts or Horn. And we simply, with so many GWAS in the portal, don't know a priori what the overlap is between these samples. And when you do have sample overlap, your test statistic becomes inflated. And again, it's not a statistically rigorous analysis. So the goal of the bottom line analysis is to kind of provide a good compromise here, to do a meta-analysis, but to statistically correct for overlap that we can observe. And as I said, this is work, this actual statistical method was done by Savanti, mainly with Gonzalo and Mike. It was implemented on the portal by Jeffrey Massling, an old engineer, and Patrick is going to take us through kind of the details of how that's implemented and how that works before I come back to show some results. So Patrick, I will now turn it over to you.
Sorry, I had to unmute first, it looked like. All right. Yeah. Cool. Yeah. Thanks for the introduction there. Yeah. So I'm back an engineer with the portal team, and as Jason said, I'll be taking you through kind of the bottom line analysis and some of the details about metal, the underlying kind of algorithm that's used there. Yeah, and as Jason also touched on, there's many different developers at the University of Michigan who worked on this and are continuing to work on this. And in particular, recently we've been working with Ryan Welsh as well, as far as some of the improvements that we can do towards bottom line. So yeah, so just getting right into it, bottom line, basically how it works is, as Jason explained, there's many different studies. There's, you know, as many as whatever, nearly 100 studies sometimes in some of these phenotypes, and they can come in several different flavors, as far as some are ancestry specific, so they can just be European only or African only. And then some are also mixed, so they can be themselves meta-analyses or kind of trans-ethnic results. And so the first thing we do as far as the bottom line is concerned is split those out, just because as far as the overlap method is concerned, you can't use the mixed results inside the overlap method. So that's the first step as far as this bottom line method is concerned. After that, we also have to split out the rare variants. So if you have allele frequency less than 5%, we classify that as rare. And that's again, just because some of the assumptions in the metal overlap method kind of require them to only act on common variants. So once you do that, you now have a set of rare variants for each of the ancestry-specific studies. And then you have a set of common variants for each of the studies as well. And those are what ultimately go into the first kind of run of metal, which runs against each of the ancestries individually. So here it'll combine, for example, just all of the European common variant results together into a single kind of now meta-analysis. And this is using with the overlap on as far as metal is concerned. And again, we still have the rare variants and the mixed variants kind of sitting. Those don't go through that process. But now we'll have 6 individual files for the 6 different ancestries that we have in the portal at the moment. And once that's done, you have to add the rare variants back in. So the way that's done is just if you have all these variants, I mean, they're a disjoint set of variants, the rare and the common. So there's nothing they have to do as far as combining those together. But if variants are across different rare data sets, then you pick the one from the largest data set and you add those back, and you basically append those to the end of these single common files to form a single ancestry specific result for each of the ancestries. And then finally, you can run metal again for the second time. This is the trans-ethnic step, and this will be with overlap off, because as mentioned, when you go across ancestries, you can't use the overlap method. So this is just the baseline kind of naive version of metal without overlap. And that forms, again, a single file that's now its own mixed kind of trans-ethnic ancestry for the overall kind of ancestry-specific results. And then finally, you have to add the mixed variants back in. So there, basically, you treat the trans-ethnic as its own mixed kind of result, and then you just take the value from the largest data set. So it can be either the trans-ethnic result itself for each variant, or it can be if there's a very large mixed data set, for example, that will end up being the representative p-value, and that forms the final bottom line result. So that's just kind of a large overarching kind of overview of how the bottom line operates. And you can see this, I think, pretty clearly on the variant page of the portal. So basically, something like atrial fibrillation, you can see just this p-value matches with one of the data sets, in particular the largest data set. But if you look at heart failure, for example, it's combined all three of these together to form its own new p-value by combining the sample sizes together. So you can see that on those pages, you can see kind of how the bottom line has treated all of the individual, and at least to some degree, kind of back calculate exactly how it's come to the conclusion it has. And then, yeah, just to step through METAL a little bit. So yeah, this is just the documentation for METAL. It's at the Center for Statistical Genetics, the University of Michigan page. And this has a really good overview of how METAL operates and how to run it. But in particular, here's just, I highlighted the section that highlights the sample overlap, because that's what we've mostly been concerned with for the last few months. It's kind of understanding and then hoping to improve the overlap method to some degree. And it has a full preprint, full method description, which I found quite helpful in understanding how all of that operated. And yeah, just to step through that a bit. So as Jason kind of touched on, if you run it with overlap off, it's a fairly straightforward method. If you have two studies, for example, and you're looking at a single variant, in each study, the variant will have a different sample size, N1 and N2, and have a different z-score, Z1 and Z2. You can just treat those as kind of a normally distributed random number. And then just to combine them, you combine them as you would any normally distributed random number. So the effective sample size will just be the linear combination of the two sample sizes, and then the z-score will be scaled by the square root of those sample sizes to form a new kind of meta z-score. And you can do that for as many studies as you want. You can do it in sequence. So no matter what order it is, you end up with the same z-score, this meta-analysis z-score. From there, you can kind of back out the p-value again. And that'll just scale again. The effective sample size then is just the sum of all the individual sample sizes. But basically, when you're trying to deal with overlap, what the assumption you're making then is, oh, there might be this smaller sample or set within the samples that are overlapping. So there's this N1, 2, and basically the question that the overlap method tries to answer is, how do you determine a good value for N1, 2 when you don't have access to the kind of underlying individual level data? And so, yeah, that's what overlap is basically trying to figure out. And just to step in a little bit more detail through the actual mathematics. So yeah, this is, again, just the original version overlap off of METAL and how it combines the two. So the question, like the way you can basically think about it is, if you have the two studies, you have the Z1 and 2, and Roman numerals will be kind of the set of non-overlapping regions, so that the independent set for study 1 and 2. But then you also have a z-score that represents the overlapping set Z1, 2. And so just using these two equations, if you multiply them together and take the expected value, N1, 2 kind of falls out. And as long as you can figure out a good approximation of what the correlation between 1 and 2 is for that variant, then you can get a good approximation of the overlap that you might expect from between the two studies. So N1, 2 then is the square root of N1 times N2 times this correlation. That is, I'll leave open as far as exactly how that is determined in METAL. And so, yeah, once you have the overlap region, the effective sample size is pretty straightforward. If you think of it like a Venn diagram, the combined then is N1 plus N2, but then you subtract the overlap region, which has been counted twice in the sample. And then again, if you just look through how you might kind of try to weight Z1, 2, what you're trying to figure out is this value W. And what basically the target that you can look at is we want Z12 to have a variance of 1. And so once you think of it that way, and you basically multiply by itself and take the expected value, you can figure out that W must equal N1 plus N2 plus 2 times this overlap region. So, and once you look at the final thing, it becomes clear that what's happening is it basically is just scaling down by increasing the denominator of this fraction. So basically, the more overlap you have, kind of the lower Z12 will end up being, which makes sense because what's happening is you were getting too much power from assuming that the 2 studies had no overlap. And so, yeah, what you end up with is a kind of new approximation based on, as long as you know kind of a good value for this correlation, you can figure out kind of better values for the combined studies, both the effective sample size and the z-scores. And ultimately, it's the same thing. If you have case studies, then it's fairly simple to extend this to a number of different studies, just by, again, knowing the correlation between each of the individual studies. You can figure out what a good meta-analysis z-score and effective sample size should be. And yeah, some of the improvements we've been working on with the University of Michigan team, in particular, just has to do with the effective sample size. So we noticed that there were some sample sizes that were seemingly lower than you might expect, to the degree that even they were negative. And so when investigating that, it became evident that what was happening was, if you think of these 3 studies that you're now trying to combine, you can have an overlap between each of the individual studies, N12, N13, and N23. But if you do the math out as metal with overlap on does, you'll find that it subtracts all 3 of those regions. But if there's a significant region in which N1, 2, and 3 all overlap, you'll end up underrepresenting this effective sample size. And so what we're working on with the Michigan team is basically just adjusting that so that instead of combining them all together in like the single step, as it were, it will instead do it kind of like a daisy chain. So for the effective sample size, basically it'll set the sample size for study 1 and 2 as you would normally do it with overlap. And then, but when it comes to adding in 3, it'll then calculate a new overlap that is between this new kind of effective sample size, 1 plus 2. And that way, it should guarantee, as long as you have good approximation of the correlation, that you shouldn't get like sample sizes that are less than 0, and it should be closer to what you would expect the sample size to actually be. Okay, there we go. And then finally, something that I've been working on is just investigating a little bit about how the correlations are calculated. So here's just an example of the output. Basically what it's doing is it's grouping these markers together based on the weight, the combined weight of the 2 studies, and it's calculating a correlation from that. And that's the correlation it will assign to each of the variants. But something I noticed while doing some of these investigations is just that some of the buckets have very few markers. So this one has 6, for example. And in the original preprint, they explained that something that might make sense is to use k-means clustering instead to try to get a better representative group for these markers. And so I've just been exploring recently. They didn't use it in the ultimate algorithm because it takes too long. But as far as like an investigation is concerned, I've been looking into basically what the consequences are of using different ways of binning these markers together. Yeah, and that is all I got. So I guess I'll hand it back to Jason.
Patrick, before you go, there's a question in the chat from Kim. How much does order of analysis matter when you daisy chain them?
That's a good question. Yeah, so we did discuss that with the Michigan team. So theoretically, the order shouldn't matter, but it does matter. And at least I think that it probably has to do with how the rows are binned and kind of combined or calculated, rather. And so yes, the order will matter. And what we're probably going to end up doing is just determining an order, at least for the portal. Probably something as simple, just like greatest to to smallest as far as sample sizes for the different studies. So yeah, order does matter. It will give you different results. I do think if depending on the binning method, I think it'll mitigate some of the differences in the permutations. But that is definitely something that we're exploring as far as improvements, trying to reduce the dependence on the order of studies. Cool. And now I'll hand it back to
Excellent.
Jason.
Thank you, Patrick. Awesome.
We have another actually, do you mind, Jason? Alex says, excellent presentation. Can you explain again, how do you calculate the correlation of z-scores between studies? On your explanation, it seems you calculate the correlation between studies, but not between z-scores in different studies.
Yeah, you calculate it. The way it's calculated is basically to take it bins them together, all the markers. So you'll have something on the order of a few hundred markers that are binned together, and then it will just take a maximum likelihood estimation of. It has a threshold. It's a more complicated than this. There's a thresholding thing to make sure that variants with very high p-values or very high z-scores don't get in there and are used for it. But basically use that to figure out what the most likely correlation is within that bucket. And then it also is a condition that it can't be negative. So it's the minimum correlation of 0.
All right, I see a question from Doug as well. How do you plan to treat whole genome sequence data sets in the future that will have far more rare variants, et cetera? So that one will unfortunately be limited towards those variants will just have the p-value from the largest data set that they are in. We will not be able to meta-analyze those. So, if you have hope, on the optimistic side, most of the whole genome data sets, the ones that are rare, will be private to that data set, so you shouldn't lose too much, but that is a current limitation of the statistics. All right, let's see from Karen. It is easiest to interpret variance in genomic region when the sample size is relatively similar across variants. How similar are the sample sizes between variants? Do all included data sets have similar genome-wide coverage, or are data such as Metabolic or non-imputed data sets included? The latter, is it feasible to include an option to display results only for variants with a threshold of some percentage of the maximum sample size? So that's a great question. And in fact, the first caveat I was going to mention as we get to talking about how this works in practice. So the heterogeneity of sample sizes is a substantial issue here if you're going to download the results from the portal and use that for like downstream post-processing methods like credible sets or things like that. And it's not just an issue with sample size heterogeneity, it's also like obviously LD assumptions and things like that will break. So we've definitely seen a lot of issues with taking the bottom line results out of the box and running like genome-wide post-processing methods with those. And that's something we're going to continue to explore. For the moment, I think that for those methods, it's far better to just simply take the largest data set at the moment. For your question, Karen, is it possible to only show variants that have above some maximum percentage of the sample size? That's certainly possible. In the portal currently, most of the modules are more focused on like, is there a signal in the region or what's the p-value for this variant? In which case, it probably doesn't make sense to filter. But certainly if you're doing something like on the new variant sifter where you're trying to compare p-values, having a filter on sample size would be a nice feature to add, which we should make a note of. One of the other things that currently makes that particular thing a little challenging is that the reported sample sizes currently have some issues in the portal. There's no concern with that in terms of the validity of statistics, but that's something that we're working on. So I guess that actually was a good segue because it allowed me to cover all of the caveats here that we had. So please feel free to follow up again in the Q&A if you have others from that. But let me get on to actually talking about whether this works in practice. Before I do that, I just want to give a kind of technical caveat that the results I'm going to be discussing are not actually of the results in the portal. And that's primarily because the way that the bottom line is currently implemented in the portal is that we run it across every single data set. And that includes data sets that are like obvious overlaps. So these data sets I'm showing in this table are actually fed into bottom line on the portal, including the male subset of giant is like meta-analyzed with the full subset of giants. And that's obviously not, that's kind of silly to do. And we're in the process of fixing that. You should rest assured that when we have the statistics still seem pretty well calibrated in this case, the overlap algorithm seems to be pretty good at detecting that. So we don't have any reason to believe the results are problematic in the portal, but just for cleanliness sake, that we're going to, going forward, remove these obvious cases, and we'll update the portal with that. And all the results that I'll cover are under that cleaned up analysis. So for example, rather than analyzing all of these traits here, we're talking only about the ones with blue here. We've removed all the ones that are obviously white. And Karen, this should answer your question too, that yes, we do include exome chip and metabolic chip data sets in here again, because our goal here is really, we're only looking at for each variant by variant, what are the associations that, are they associated or not? We're not at the moment concerned with how to look across variants and handle the heterogeneity. That's something we're going to do in the future. Okay, so the first question that I think is warranted is, how many traits is this even an issue, right? Obviously, if there's a trait that only has a single data set, this is all moot. So it turns out that about 28% of the traits in the portal have at least 2 data sets for them. So this is an example where in principle, some meta-analysis could be potentially useful. And then when you start binning those traits, you can then classify them into roughly 4 different scenarios. So the simplest scenario is when there's just at most 1 GWAS per ancestry, so 0 or 1. And there's no multi-ancestry GWAS. So it's like a pretty straightforward, like transethnic meta-analysis where no overlap detection is needed. Slightly more complicated would be you also have a multi-ancestry data set. And again, this is a simple scenario where you're essentially just like meta-analyzing without overlap of the ancestry-specific ones, and like potentially taking that or the mixed data set, depending on which is larger for a variant. Scenario 3 is when things get interesting, which is what, as Patrick alluded to, within an ancestry, for at least one of them, we have multiple data sets. So combining these requires the overlap detection algorithm to work. And that could, there's slight differences whether you also have mixed data sets or not. But for the purposes of today, we do not yet have any results for these first 2 scenarios. This is something though that we want to look into because, the ability to do transethnic analyses with data that haven't been previously combined is probably noteworthy. But today I'm going to be focusing really on scenario 3, because our really goal here is to try to evaluate how well this overlap detection algorithm is working. So from that matter, we're not just considering trait by trait, but actually trait ancestry pair, right? So like you might apply the bottom line analysis with overlap on for BMI and 5 different ancestries here, right? So if you look at this table here, we have 2 data sets for African American BMI, 4 for East Asian, 12 for Europeans, and 2 for Hispanics. And so these are 5 separate analyses we'll be discussing. And this probably kind of gives you just a landscape of how many data sets we're dealing with here. Once you remove all the obvious overlaps, the modal value is like 2, right? But there are some traits that, and particularly the ones like blood pressure and BMI and height that are measured in so many different studies, that we have tons of different studies that in principle we need to think about how to combine. So here's the setup for how we evaluated these. For each of these pairs, we're conducting 3 different ways of combining the results. That minimum p-value approach I mentioned earlier, what we're calling the naive meta-analysis, which is just running meta without any overlap of detection, and then the bottom line step, which again, just the overlap, aware meta-analysis here. Then what we do is we clump each set of results into independent associations. So, this is the goal. If you haven't thought of clumping before, it's just if you have 10 variants associated in a region, it's just kind of merging them all together if they're in LD. So you're trying to reduce to the set of independent associations that you have. And then what we're trying to do is determine how many traits, answer questions like, is meta-analysis potentially profitable? How many associations we're gaining or losing, and how accurate the new associations are that we detect. And just the way we consider an association to be found in 2 studies is if the clumps overlap at all. So if the minimum p-value has a clump of 10 variants and the naive meta-analysis has a clump of 10 variants, and they share even 1 variant, we consider that to be catching this exact same signal. Okay, so here's just to start with kind of an upper bound on what you can expect to gain by these analyses. So I'm plotting the sample size of the largest data set for each trait ancestry pair. Those are the black dots, and then the sum of all of the sample sizes, which kind of gives you an upper bound on the sample size increase if there were no overlap. And this is log scale, so you can see that for most, the expected increase is somewhat minimal, but you do have some where you have an order of magnitude gain in sample sizes here. In this case, I think from 5,000 to 50,000, for example. And then kind of, if you want to think not just sample size, what's our upper bound for gain for that, but what's our upper bound gain of the number of associations that we might add or lose based on this analysis? What we can do is compare the naive meta-analysis results to the minimum P results. So another way to think of the minimum P value results are that we consider a variant associated if any data set has it as associated, right? And then naive meta-analysis is kind of like if we fully meta-analyze them, would we find anything new? And what I'm showing here in the red bars are the associations that are gained for each trait relative to minimum P and the associations that are lost. So you can see that overwhelmingly, the meta-analysis is adding new associations for many of these traits. Again, this is the naive one, not the bottom line, but it's also pruning out some associations in white, although that's smaller. And those presumably are associations that are found in one data set, but don't replicate in another data set, right? And so that's kind of the landscape of what we could hope to find. So now digging into what the bottom line algorithm actually does, right? So relative to the minimum p-value, you can see this plot looks quite different, right? The red bars are way smaller, and the white bars, particularly for certain traits like triglycerides and cholesterol, but also for the scale is much higher here, right? This goes to 200 rather than 100, are pretty large. And the numbers overall is that 6,644 associations are shared. It gains 346 and it loses 895 relative to the minimum p-value. So it seems to primarily function by pruning out associations that are significant only in one data set, but you do see some novel associations identified, a small number. Now, relative to the naive meta-analysis, it's actually interesting. It gains fewer, but prunes out far more. So essentially the way to break these down are that if you run the bottom line analysis relative to the minimum p-value, it gains 327 associations that the naive meta-analysis would also detect, but it finds a lot more that are the naive meta-analysis thinks are accurate, but are not accurate once you correct for overlap. But it actually even gains some associations relative to naive meta-analysis. And we haven't yet picked through exactly what the properties of these are, but I think the overwhelming message here is that the primary function of the overlap or analysis is to actually prune out associations, kind of straightforwardly you would expect from the naive meta-analysis, but also the minimum p-value analysis, although there is some gain of associations as well. In terms of looking at, those are just raw numbers, are the results, do we trust the ones of the bottom line to be better than the other methods? So this is a plot of the lambda values for each data set, each trait for the bottom line relative to the largest data set. So the largest is a QC'd cleaned data set we're taking in. And you can see if anything, the bottom line is conservative. Lambdas are smaller for it on average relative to the largest. And for those who aren't familiar with lambda, it's a measure of how well calibrated your statistics are. It should be 1 if your data set completely follows the null, and the larger it is from 1, the more you're seeing deviation from that, which in many cases we think are due to artifacts in the analysis, although for reasons of polygenicity, you don't always expect the value to be exactly 1. So at least it certainly doesn't look like we are inflating the statistics globally unreasonably. And it's and these are actually two interesting plots now comparing it to the minimum p-value analysis and the naive meta-analysis. So the minimum p, you can see this just kind of validates again that it's not a really viable strategy here. If you actually look at this statistically, the lambdas are just completely unreasonable. What is interesting actually is that the naive meta-analysis looks pretty good, at least based on this metric, right? So for the most part, the lambdas are quite similar between the naive meta-analysis and the bottom line, which I guess on the surface would suggest the overlap of these data sets is at least not showing up in the lambdas, which will be interesting in a moment when I show you the validation results, because I think it's not obvious to me why the naive meta-analysis appears to be so well calibrated relative to the bottom line. Okay, so the final analysis now is trying to figure out a different way of evaluating whether the associations that we gain are real. Ideally, we'd like that the associations gained by the bottom line analysis replicate or are expected to replicate more so than the associations that it filters out, right? So to do that, we did another experiment where we first took the largest data set for each trait and held it out as a validation data set. So for hypertension, this data set from 2019 with 460,000 samples. We're taking that out completely, and that's our gold standard data set. Then we rerun the bottom line and the minimum p-value and the naive meta-analysis for the remaining data sets that don't obviously overlap. And then we do our previous analysis where we identify the signals that we find in each of these three methods, minimum p-value, naive, and bottom line, and ask how many of those, what fraction of them, are found in this larger data set. So we're kind of trying to do we're pretending like we ran this 3 years ago before this study was done, seeing what we found, and then asking how often are those associations going to replicate, and hopefully it's going to be a larger number for the bottom line than it is for the other methods. So here are the overall statistics. Again, remember, this is not the original results because we're running this in this kind of fake validation scenario, not fake, but the reduced one. So the bottom line finds 2001 associations overall, and 81.4 of those replicate in the larger data set. And this is kind of a scatter plot of each trait ancestry pair. You can see that there are some, obviously the majority of the data, there are some data sets traits where we find a large number of associations, but you do see consistently high replication across most of the pairs. Although, we'll have to look into what's going on here. In many cases, these are just, only a handful of signals anyway. Now, compared to other approaches, this is I'm showing the bottom line replication rate on the x-axis now, and the y-axis is the replication rate of either the largest data set, the naive meta-analysis, or the minimum p. Now, remember, in this case, the technically largest data sets are validation set. So largest in this case means after we removed the replication or validation data set, what was the largest one remaining? And it's quite notable that the replication rate for the bottom line is larger than that largest and substantially larger than these other two approaches, which is probably what you would hope to see. And the number of associations for the bottom line is actually lower than the largest here, but given that the higher replication rate, there are more associations that are actually replicated that it finds. The naive meta-analysis finds the most associations, but the replication rate is so low that you end up, this comes at a cost of a much lower validation rate. And what we'll be doing in the coming weeks is kind of, so basically the right-hand side of this plot are those where the bottom line has a higher replication rate for a given pair. What we'll do in the coming weeks is kind of look up here to see if there's anything particular about these trait ancestry pairs that might indicate why the bottom line appears to be not doing as good of a job as these other methods here. So to kind of conclude with the bottom line of the bottom line, if you kind of add everything up, there's a couple hundred associations that it identifies that are not present in any individual GWAS in the portal. It does remove a much larger number of associations that are present in data sets, but don't have supporting evidence from the others. And the naive meta-analysis, despite having what seems to be a pretty good lambda value, is not a viable solution in this case. The overlap really does result in a large number of what we think are false positives based on our validation analysis, a much lower replication rate. And kind of, I'll kind of wrap up there. Like I said, we're as I kind of alluded to throughout, we're kind of working now on further analyzing these results. And as I mentioned, we're going to be using some of these findings to help clean up and improve the process of the portal. And as Patrick mentioned, we think there's still some room for some further statistical improvements to the method that we'll be working on together with Patrick taking the lead, but also working with our colleagues at Michigan. So that I'll turn it back to Noelle.
That's great. Actually, we have a couple of questions in the chat. I have one too. So do you want to read that one out?
So Kim's question, are the lambdas from the version that includes the obviously overlapping results or the new analysis that excludes those? So the lambdas I showed are all based on the new analysis that excludes those. We do have, in the past, we have looked at lambdas for the ones that include the obviously overlapping results. And I don't have a systematic comparison, but they don't appear substantially inflated. Like we still, if anything, they're conservative in that case. So like that was the reason I gave that caveat at the start. Ideally, we would, we're going to improve we're going to eventually make the results I showed concordant with what's shown on the portal. But the ones in the portal, from everything we've seen in the data I didn't show, we don't think there's a problem, at least based on the lambdas that you're seeing.
I have a question. So, because this is very unique to our portal and it's not transparent to the user right away, what new results might be in the portal based on what we've done of analysis behind the scenes. Can you speculate as to how we might represent these results on the portal in a way that users can quickly access and sort of distill when they're looking at a given page?
I guess probably if your goal is to like allow users to navigate to only identify the novel associations, that I guess could be done in two ways. One's on the phenotype page where you are browsing the top results. You can imagine that it's separate, the ability to have a new card that only will show the novel ones. I could also imagine maybe propagating for each variant, like, is this, in which combination method is this significant, and kind of leveraging that in the UIs. And maybe you could allow filters there. So like on the locus page, you could say like, remove all the variants that don't show evidence of association in both the bottom line as well as the individual data set.
Yeah.
And that could be something we could do. I think it would be interesting to think about exactly, what the use of that is. Like, I guess Karen's question kind of relates to that, right? Which is you could imagine that there could be scenarios where you want to filter down to variants that have, specific properties, like have to be observed in X percent of samples. And that could be something we could think about propagating, so we could add that functionality.
I say this because of two reasons. Most of our citations come from people using the portal as they look up for their favorite variant or gene. And so having some provenance in terms of what those novel results are is really critical. And then the other thing that I think would be really kind of cool is you can imagine using our BR framework, a plug for our software, to almost have a separate page in the portal or a separate module that says results from bottom line method, and literally just having like a chart, like almost exactly the chart you displayed, which is for all the phenotypes, traits in the portal. You have the number of data sets and the number of signals that are identified from the largest data set, and then it was an interactive table, and then sort of the delta based on the bottom line of things we exclude versus the things we include, and then a table below that filters based on those. And then the user can then go to that page in the portal. You can imagine that being a very useful
that would be cool.
tool. That's what I was thinking about when I saw these results, because I'm particularly excited by these, because this is a really cool feature for our resource. So with that, any other questions on the bottom line before we convert to Maria's tour of our new data and features?
Just one final, just again, thanking Firkin for most of the analysis that we shared.
I think he's on the call. Thank you, Firkin.
And Patrick for all his work on improving the method, and obviously Sabanti and Ingasal and Mike for developing it in the first place. Yeah,
it's pretty cool because it's the ultimate display of the collaboration that I think the consortium conveys. It's bringing all the data sets from our contributors, our community, publicly available GWAS, pulling those all together, implementing novel methods, creating visualizations, and validating them. So pretty cool. Anyways, with that, Maria, would you like to take the helm for a little bit?
Sure. I'll give a lightning tour of our latest release, which we did in June.
We're good.
Let's see. Why is this not responding? There we go. Okay. All right. So this slide is, you're not meant to read this, but I just want to make the point with this slide that we have really ramped up our loading of genetic association data sets. So this is awesome. We were able to add 51 new data sets this release, representing 13 traits that are new to the knowledge portals and a whole bunch of traits that were already represented there. And this is mainly due to two reasons. One is that our collaborators at EBI are really familiar with GWAS catalog, and they've been helping us to download selected data sets and harmonize them before sending them on to us. So that really speeds up our loading of them. And then also our computational biologist has developed more automated ways of dealing with and loading the data sets on our end. So with those two things combined, we're really zipping along. And there are so many data sets, I don't think I have time to really discuss each one, but I'll give a few of the highlights. So one of the really long anticipated data sets that we've just loaded is the largest to date study of T2D from the DIAMANTE consortium. Almost, I think 1.3 million people. There's a trans ancestry meta-analysis and also some ancestry-specific sets. And we also have the credible sets from this analysis, and I'll talk a little bit more about those later. We're also hosting the summary stats and the credible set data for download for these data sets. We've added some new glycemic trait and data. So we have a MAGIC consortium study of sex stratified results for fasting glucose and insulin. We also have a study of youth-onset type 2 diabetes from the ProDiGY consortium, and that's available for download as well. We've added a large new adiponectin study, also a study of diabetic nephropathy in East Asian individuals, and a study of coronary artery disease as a complication of type 1 diabetes. So for type 1 diabetes, we've added one new data set, and it's very small, but it greatly adds to the genetic diversity in our portal because it's from the Amhara population of Northwest Ethiopia. So increasing the diversity of our samples is a constant goal for us. And this brings us towards that, and where we certainly want to continue to increase the diversity in the future as new data sets become available. We're hosting the summary stats for that study. And then we've also added a whole bunch of allergic and autoimmune disease results to kind of accompany the T1D portal. For cardiovascular, we've got a huge new study of blood pressure in 1.3 million individuals. We've also loaded a mitral valve prolapse GWAS study, and we have both the summary stats and the polygenic scores available for download from that. An imaging study of the left ventricle, and a study from TopMed of several electrocardiogram traits. And we have those summary stats as well. Plasma lipids, we've got another leptin study, and we've also loaded a 170,000 sample exome sequence analysis study of plasma lipids that has a lot of ancestry stratified sets. And for that one, we have both the summary stats and the gene level association scores available for download. Some miscellaneous traits, we've loaded a study of height from African-American individuals. We've also got the largest to date study of gestational diabetes. And we've loaded a study of the progression of age-related macular degeneration. So we've also updated our exome sequencing results in the portal. So you may recognize the AMP T2D-GENES. This consortium did exome sequencing of about 50,000 samples. And we've had the type 2 diabetes results in the portal for a while, and also some of the quantitative traits from a subset of these individuals. These were called the 13K exome data sets. But now we've added the associations for all of these 24 quantitative traits from all these different ancestries from this data set. And these replace the 13K from before. You may know that we have added a new portal recently for non-additive genetic effects. So these are associations determined with other models besides the additive model. We just added a new study of T2D associations to that portal that has both additive and recessive model associations. Let me just check my time. Okay, a couple more minutes. Okay, and as I mentioned before, we have credible sets, new credible sets. So we have them both for type 2 diabetes from the DIAMANTE study, and type 1 diabetes from the largest T1D GWAS from Kyle Galton's group. And so these are both, manually curated sets, and they're now available both through the GEM genomic region minor module on the region page, and also through the variant sifter. And here's an illustration of what they look like in the variant sifter. So these on the top, these are associations across the region of block 2 for type 1 diabetes. And then here are two different credible sets from the Kyle Galton study. Okay, we've also changed our FIWAS displays a little bit. So we are FIWAS plots that are on the region and variant pages are more interactive now. More interactive and more informative. You can see the direction of effect for each association, and you can also, if you mouse over, you can see all of the information about these associations. And we've added a new FIWAS plot of rare variant gene level associations to gene pages. Okay, and finally, some of you have participated in a focus group about this new in development tool, the non-coding genetic association interactive tool. So we've now added a link to it to our production site. It's in the KP Labs menu. Very briefly, how this tool works is that you start by selecting a gene, a data set, and a phenotype. Right now, the only data set is from TopMed and it's T2D associations. But we're planning to add more data sets. I think the next one will be from UK Biobank. You then select one or more annotation types and tissues, and this global enrichment plot is shown, and that will give you an idea of, for the phenotype you're looking at, in this case, T2D, in which tissue-specific annotations are these associations enriched. That can help you in choosing the tissues and annotations that are of most interest. And then you can select multiples, and you can decide to look at their union or their intersection. It's up to you. And when you settle on the regions that you want to look at, then you can retrieve the variants that are located within those regions. You can further customize the list by checking or unchecking individual variants if you want. And then you can choose to run any or all of 4 different aggregation test methods to see what the burden is for association with the phenotype of choice across the regions that you've selected. So that is all that I have. Any questions about that? I don't see any questions.
Maria always makes these excellent videos. So if you missed any of that, she will post that shortly to the portal for you to peruse. I'm going to shorten this, but I want to highlight 2 things that are coming up this summer. Specifically, as you see from the data sets we're trying to incorporating, we're trying to address all of common disease. And we're very lucky that the NHGRI thought that our platform that we had built for diabetes and common metabolic disease was really important to the community, and therefore they funded us to expand this resource to all of common disease, titled the Association to Function Knowledge Portal. And what I wanted to at least preview for you today, and we'll leave you with this, is that in July, we'll be July 14th at the same time at noon for our webinar, we'll be touring you the beta site of the A2F knowledge portal. What's nice about this is you can tour it now if you want. It includes all the data sets you see in the CMDKP, as well as all the community portals that already exist. It allows you to tour all the exact same features and tools that you've seen here, but in one framework for common disease. But also this brings our new community portals, an opportunity for us to connect with disease communities that do this better than we do and understand the disease data sets and the traits, cell types and the data sets better. And one I want to introduce today is the neurodegenerative disease knowledge portal. We have a beta version of that out now, and we'll be talking about that in July. So I invite you to join us to that webinar. And then in August, Klaus Kessner is going to represent the AMP CMD consortium and talk about his results working with the Penn team on effector genes for type 2 diabetes and common metabolic diseases. So look for that this summer. And again, if you miss any of this, it'll be on the webinar. Oh, I see a chat. Sorry, let me grab that. Oh, great new data and tools. Thanks, Melissa. That's nice to hear. So if anyone has any questions, feel free to email us afterwards. Thanks for tuning in. Thanks to Jason. Thanks to Patrick, and thanks to Maria for presenting today. It was really great to see these results, particularly for the bottom line. I'm very excited. And I look forward to seeing you all again, and have a great summer. Take care.
Right on time.